Okay, it's about time that we actually start talking about diseases by body system. And we start with chapter 19, the diseases of the immune system, because we just completed going over the host defenses in both the innate and adaptive immune responses. So we'll start with the immune system, and then we will move into other body systems. I mentioned a long time ago that I like the way that this book is organized with the diseases by body systems, because in any given system, the way that the pathogen has to evade the host defenses is going to be similar. And so we will look at bacterial and viral and fungal and other pathogens. So there'll be a diversity of pathogens in each of these systems, but they will have to accomplish similar tasks to be able to establish themselves in that body system. So diseases of the immune system can be quite dangerous because they are attacking the very system that we have to defend ourselves against pathogens. And some of the diseases we're going to talk about have to do with allergic responses. So that's why we have an image of a bee and an EpiPen. So bee stings are a particular kind of event that can cause an allergic response in many people. The venom in the bee sting is a protein that can trigger an allergic response. And in most people, it's low level and painful and irritating, but that's about it. And other people, they can have an entire systemic response. So we will talk about what that response is and why an EpiPen that contains epinephrine is the antidote to that hyperimmune response. It counteracts that drop in blood pressure because your non-specific immunity that is happening initially causes that vasodilation that causes the blood pressure drop, and epinephrine can counteract that. So different allergens shown here. Pollens. When you see an electron micrograph of pollen, you can see why they might be extremely irritating to your respiratory passages. Allergic rhinitis is inflammation of your rhino, your nose. So rhinitis is inflamed nasal passages. Also called hay fever, because hay is often a trigger of allergic rhinitis. This person is suffering a contact reaction to some sort of allergen. And then we have peanuts, a known severe food allergy. And food allergies can be some of the most dangerous because it doesn't take much of that allergen to trigger a whole body and severe response. A lot going on here. This is showing how allergies can become more and more dangerous. In our specific or adaptive immunity chapter, we talked about initial exposure to an antigen, and there is a specific response that then declines. And when there is a secondary or tertiary or quaternary additional exposures to that antigen, there is a very rapid and much stronger response. And that is how the immune system should work. But with allergens, an allergen is a hyperimmune response. Your immune system reacts way above and beyond what is necessary. So if there's some antigen that gets processed by an antigen-presenting cell, that means we grind this up, and the class 2 major histocompatibility molecule binds to that antigen and presents it, shows it on the surface of the antigen-presenting cell. That means that the helper T cells that have the receptor, the antigen binding receptor for that, will bind to that antigen. Helper T cells release the cytokines that activate the B cells. The B cells are the cells that make antibodies. And so they proliferate, which just means divide, multiply, and differentiate or mature into plasma cells. And they are secreting an IgE antibody, immunoglobulin E, that is specific to that antigen. And then those plasma cells will bind to mast cells by the Fc region, the antibodies. The little forks, the tines of the fork are specific to the antigen, right? They're the variable region. The handle of the fork is the constant region. These antibodies bind by the constant region to mast cells, and when mast cells are triggered by that, they secrete inflammatory molecules. And because the antigen binding portion is still available on these antibodies, any subsequent exposure to that allergen, that antigen, increases the inflammation response. And that is why the first time you see an allergen, you might have a mild response. Secondary exposures, you might have a additional rapid and stronger response. And food allergies definitely work this way. There may have been a mild response early on that wasn't noticed, and the second response, we have all these sensitized mast cells that secrete an abundance of inflammatory molecules and cause that full body reaction. This is looking at our blood types. And our blood types are classified according to the antigens that are on their surface. Our blood cells definitely have MHC class 1, that's on all of our cells, but they also have specific antigens that identify them as a particular type of red blood cells. So type A has A-shaped antigens. Type B, we have little squares instead, so that is the B-specific antigen. Type AB has both. And type O has neither lollipops nor squares. It doesn't have either. It has other antigens on its surface that identify whose blood it is, but it doesn't have A antigens or B antigens. And because you have particular antigens on your blood cells, that means you have specific antibodies against other blood types. So if you have type A antigens, then you make antibodies against type B antigens. And the specific antibodies we're talking about are IgM, because they are those pentamers. They have 5 little forks that make them up. They are called isohemagglutinin. Heme means blood. Glutenin means will cause it to clump. It's glutenin, not gluten, right? Gluten, to agglutinate, means to clump. And iso means similar. So they're similar in shape, but they're going to cause a blood cell, which is a similar type of cell, but has a different antigen, and will cause them to clump. So A antigen folks have anti-B antibodies. B antigen folks have anti-A antibodies. Type AB folks have neither anti-A nor anti-B. They have other antibodies, but they don't have any isohemagglutinin antibodies. And then persons who are type O have both anti-A and anti-B isohemagglutinin, because they don't have either antigen, so they will have antibodies against either antigen. And this deals with blood types and whether you can donate to another individual and what types of blood you can receive. So if you're type A blood, you could receive type A blood, right? Anti-B antibodies aren't going to affect you, and having A antigens, you're not going to make any anti-A antibodies. Likewise with type B blood, you could receive type B because you don't have any A antigens, you only have B antigens, it's okay. If you're type AB, you could receive type B blood because you don't make any antibodies against type B. You could also receive type A blood because you don't make any antibodies against type A. If you're type O, there's no antigens on the surface, no blood cell specific antigens. There's neither A nor B. So type O is called the universal donor. Without antigens on its surface, it doesn't matter if I have type A. If I have anti-B antibodies, those aren't going to attack type O red blood cells because there's no B antigens. If I have type B blood, I could receive type O because I have anti-A antibodies, but there are no A antigens on the surface. So type O is the universal donor. It's typically type O negative. We'll get to the positive and negative in a moment. And AB negative is the universal recipient because they can receive any blood type. The hemolytic transfusion reaction is what we were talking about. Which blood types can you receive? So if the recipient has type B blood, and if they received type A blood as the donor, type B persons have B antigens, but anti-A antibodies. And those antibodies would clump to the type A blood cells and cause them to agglutinate. When the antibodies are bound to antigen, in addition to clumping the cells, that antibody to antigen binding triggers those complement proteins. Remember that series of reactions that gets you to the C, 6, 7, 8, and 9. That's the membrane attack complex and destroys the donated blood cells. The RH factor, is there a good place to put me? Maybe over here. It's called RH because it was discovered in rhesus monkeys. However, rhesus monkeys like us are primates. We have a similar factor on our red blood cells. So this is where the RH positive and RH negative part of your blood type comes in. Everybody is type A or B or AB or O, but within each of those four categories, you can be positive, meaning your red blood cells contain the RH factor on their surface, or you can be RH negative, meaning you lack the RH factor. This becomes an issue particularly with expectant mothers. If the mother is RH negative, that means her red blood cells do not have an RH factor on them. That also means because she lacks an RH factor on her red blood cells, she makes antibodies against the RH factor. For her own self, that doesn't really matter. It does matter if she were to receive blood, it should be RH negative, because if she receives RH positive blood, her antibodies would attack that. It's also important with the fetus. If the mother has an RH negative blood type and the father was RH positive such that the fetus ended up with an RH positive blood type, the mother and the fetus are connected through the umbilical cord for a blood supply and antibodies. So in the first pregnancy, it usually isn't a problem because during the pregnancy, the mother will develop anti-RH antibodies because she gets exposed to the RH positive antigens on the fetus because the cells do mix. That fetus typically will be okay and will be born perfectly normally. However, if the mother has a second pregnancy with an Rh positive fetus, then those antibodies and then their T memory cells will trigger that secondary immune response, which is very quick and very active. And those anti-Rh antibodies will cross the placenta and attack the blood and destroy the fetal cells. And there were, in the past, many, many miscarriages because of this. In fact, in my family, there are 6 years between my older sister and myself. And it wasn't until, well, after I was born that we were able to figure out how to prevent this blood type difference from causing spontaneous abortions, a miscarriage. So my older sister is Rh positive. And the miscarriages my mother suffered between my older sister and myself, they weren't tested, but in all likelihood were Rh positive fetuses. I have Rh negative blood. My younger sister has Rh negative blood. And we were able to have the fortune to escape this effect. Now, in women who are pregnant, their first pregnancy with a potential Rh positive fetus, the mother gets what's called RhoGAM for the RHO for the rhesus factor, immune globulin. So she receives antibodies to the anti-Rh antibodies. So if she has any anti-Rh antibodies circulating, she can receive an injection, a passive immunity, because she does not make the antibodies against the anti-Rh. She receives already made antibodies to the dangerous antibodies that are circulating. So the RhoGAM injection can protect the subsequent Rh positive fetuses in an Rh negative mother. Type 3 hypersensitivities are due to immune complexes, and that is kind of what we saw with the agglutination reaction, where instead of a single immunoglobulin G antibody binding with an antigen, we get cross-linking and multiple antibodies binding to the antigens that cause this clumping. When that happens, the Fc receptors, the constant regions, bind to white blood cells and trigger that release of the cytokines because it activates the complement, causes inflammation, and can affect all of these different areas of the body. Blood vessels, because these clumped up antibodies can actually clog up and obstruct capillaries. If it's in the bloodstream, it definitely is going to affect the heart. It can get into the lymphatic system and get into our joints and cause problem. It can show up on the skin, and it definitely shows up in our kidneys because that is our filtration system. It can be, got cut off here at the bottom, a complication of strep infections, so strep throat. Most of the time, your body mounts an immune response, you get some antibiotics, you clear it up just fine, but even if you recover just fine, a second exposure, particularly to another strep antigen, can cause these immune complexes, and they tend to either clog up our kidneys or they show up in our skin because the antibodies cross-react with keratin in our skin. So there's a protein on the surface of strep that the shape of that antigen is very similar to the shape of keratin. Dermatitis, most of us will experience at some point to something. Remember that ITIS means inflammation, and derm or derma means skin. So the hapten antigens, the small little pieces of the antigens in poison ivy, or where we are, poison oak, triggers type 4 hypersensitivity. Where the antigen crosses our skin, it gets picked up by dendritic cells. That presents the antigen to a helper T cell that then becomes a memory helper T cell. We get sensitized so that if we have a second sensitization, those memory T cells release the cytokines that trigger our macrophages and the cytotoxic T cells. So that hyperinflation, the blistering, and all the other things that we see with those sorts of hypersensitive reactions. So the lesion, which is usually blistery, persists until the allergen is removed. So that's why when you're exposed to poison oak, it's really important to wash it off so that none of that oil remains on your skin. Because as long as that oil remains on your skin, that contact dermatitis will persist. The TB test stands for tuberculosis. This is a picture of an older form where the antigen, a small protein from the surface of the tuberculosis pathogen, is jabbed under the surface of the skin. If you've never had or been exposed to tuberculosis, you won't have antibodies to it, so there will be no reaction to the antigen. But if you have been exposed, then the introduction of these tiny amounts of the antigen will cause an antibody response, and it'll be a secondary response, so it'll be quick and it'll be pretty intense, and there will be a lesion that will show up because of that. This is looking at hypersensitivity pneumonitis, inflammation of the lungs. So there's poultry workers' lungs because of the pathogens that are associated in the feces of the birds. And then we have cheesemakers' disease, where the different types of mold that are being used to actually ferment the cheese get into the air, and inhalation of those spores cause an immune response of inflammation in the lungs. So there's 4 different types of the hypersensitivities. So types 1 through 3 are mediated by B cells or antibodies, and type 4 is just with T cells. So what triggers them? Soluble or dissolvable antigens trigger type 1, which means immunoglobulin E is released. So hay fever, anaphylaxis, it's a full body response. Drug allergies are a type 1. Type 2, cell bound. So the antigen is stuck to a cell. The example we saw of that was the blood transfusions or the hemolytic disease of the newborn, because the antigens are on the surface of those cells. Type 3, soluble antigen again, meaning it's not bound to a cell. And that was where we saw the clumping, where we had all of the antibodies sticking to antigens and causing clusters. And I mentioned the post-streptococcal glomerulonephritis. That's where the antigen-antibody complexes stick in the kidney, the glomerulus of the kidney, or in the joints for arthritis. And type 4 is mediated by T cells, and it can be triggered by either circulating antigen or antigen stuck to a cell. And that's that contact dermatitis. It's also seen in type 1 diabetes. That's an autoimmune response. And even multiple sclerosis. This is the joy of an antibody or an allergen test. It's a skin prick test. The numbers just are for the reference of the technician to know in what order they put which antigen. They put a drop of a specific antigen on the skin, and then they take this tiny little fork and jab it into the surface, and then see if you react. There's the wheal, which is the raised bump of the hyperimmune response, and the flare is where the inflammation spreads away from where the injection site was. So this person is got a whole bunch. These ones, nope, not too much, but there's an awful lot of reactivity going on in some of these. I had this done when I was in graduate school. We were right next to the medical school, and there was the opportunity to participate in medical experiments, and they were doing allergy testing. So I went in and got tested on one arm for various grass pollens and the other arm for various tree pollens. And as they're putting the drops on my skin, nothing happened with the tree pollens, but before they even stuck me, all of the grass pollens got a wheal and were beginning to flare before they stuck me. And so I didn't let them stick me unless they had an EpiPen on hand. I did not need it, but I had wheal and flare all over one arm for a couple of weeks. This person is suffering from a goiter. It's hypertrophy, meaning overinflammation, it's too big, of the thyroid. It should be a very small gland in the throat. It's a symptoms of Graves' disease and Hashimoto thyroiditis. And the immune system is reacting against the thyroid tissue and causing this inflammation and edema and fluid buildup. This person's eyes is also a symptom of Graves' disease, exophthalmia or Graves' ophthalmopathy. The immune system is attacking the muscles and the fatty tissues around the eyes, making them swell and get pushed forward. Addison's disease is an autoimmune disease affecting the kidneys, and it causes this reddening in the hands. It's hyperpigmentation. It's not necessarily leakage of the capillaries, but it is causing reddening of the hands. Myasthenia gravis has to do with muscle contraction. In a normal cell, we have release of acetylcholine. There are receptors on the muscle cells that tells it to contract. But with myasthenia gravis, the nerve cell secretes the acetylcholine in a normal manner, but the person's body is secreting antibodies against the receptor for acetylcholine. And if the antibodies are sitting here, they are blocking the ability of the acetylcholine receptor to receive the signal, and the muscles cannot contract. Another autoimmune disease, is rheumatoid arthritis. It's a specific type of arthritis, and it may or may not be age-related. It can happen to someone at any age, as opposed to osteoarthritis, which is usually a result of aging. This person does happen to be older, but it can happen in much younger people. This happens when your body makes antibodies to the joint tissue and causes inflammation and then distortion of those joints. Lupus, systemic lupus, is an autoimmune reaction, antibodies made to your own chemicals, and in this case, it can be the DNA and or proteins. And it affects the body all over. That's why it's called systemic lupus. But one of the initial ways to diagnose lupus is what's called the butterfly rash. It makes this reddening across the nose and down the cheeks. But the disease itself, the antibodies cause inflammation of the heart muscle, there's poor circulation, and it does cause muscle aches because it is creating inflammation in various tissues. And I think this is our next to our last slide. This is this little boy named David Vetter. It was called the Bubble Boy. It's called SCID, which is Severe Combined Immunodeficiency. In the bubble, to prevent any possible interaction with any sort of antigen, which almost anything is an antigen because he doesn't make either T cells or B cells, which means he has no specific immunity. He had only innate, non-specific immunity.